question is, which instrument helps us measure the exact weight of powders, liquids or chemicals, sometimes as tiny as 0.01 gram?
It is a weighing balance. It is also known as an analytical balance, and it has a very sensitive pan inside the glass box. It is used to stop the air from moving it. If the weight is wrong, the whole experiment went wrong.
And now the second question is, we want to check how cloudy or dirty the water is, that is called turbidity. Which instrument in our lab measures turbidity by looking at how the light scatters?
It's the nephelometer. A bright light shines straight through the sample. If one water is clean, light goes straight, little scatter. If particles are there, light scatter in all directions. The nephelometer detects the scatter light at 90 degrees.
The third question, which instrument makes samples glow or shine under special light? We use it to find tiny amounts of vitamins, medicines or DNA.
It's the fluorimeter. Shine strong light, excitation light on the sample. Some molecules absorb the light and get excited, jump to higher energy. They quickly fall back and give out their own light fluorescence, usually a different color, longer wavelength. The fluorimeter measures only this emitted glow at 90 degrees to avoid the original light.
The next question, which instrument can test blood or serum for sugar, cholesterol, liver or kidney function, all automatically in just minutes?
It is the biochemistry analyzer. Put the sample plus chemicals in small wells or cuvettes. The machine mixes them into a color change happens based on the substance. Light passes through, measures how much color absorbed. Computer calculates and prints results for 20 plus tests at once. It saves time and reduces errors compared to doing everything by hand.
Okay, now a quick question. Which one checks water cloudiness?
Nephelometer.
Correct. Which instrument is used to purify water?
Distillation unit. Laboratory and industrial apparatus use பண்ணி செப்பரேட் பண்றதுக்காக purify or concentration liquid வந்து mix பண்ணுவாங்க based on difference on boiling point.
Okay, we can dry and sterilize our sample using this instrument.
Yeah, hot air woman is an electrical laboratory device that uses dry heat, typically between 50 degrees Celsius and 250 degrees Celsius, to sterilize, dry or bake materials, including the glasses and the metal instruments. It works via connections with the heating coils and fans, providing uniform temperature distributed to kill the microorganism, generally through protocols like 170 degrees Celsius for 30 minutes.
As we can see, various questions like applications were asked to students, and they have answered everything clearly with the working principle of that particular analytical instruments. We have talked about various analytical instruments like weighing balance, nephelometer, fluorimeter, biochemistry analyzer, distillation unit, as well as hot air oven. So first of all, weighing balance is something that is used to measure the weight before or after we got our product. We have to weigh our sample before taking it for analysis, and after completion also we can measure how much amount of product that has been formed or how much the purity we have obtained. So for those purposes we need weighing balance. Weighing balance are available in various sensitivities, and each one is weighed appropriately using that instrument. Then the second instrument is nephelometer. So nephelometer is particularly used for checking the turbidity of solution. So nephelometer is a part of nephroturbidimeter actually, but this nephelometer is used when the suspension is having very less turbidity, and the turbidimeter is used when the solution is having higher turbidity. So with the help of this instrument we can measure the light scattered by the particles present in our solution, and with that we can indirectly measure how much particles are present in the suspension. Then the next instrument is fluorimeter. This fluorimeter is used when you have a fluorescent compound as a sample. Suppose if your compound is a fluorescent one like quinine sulfate, this compound have a nature of reflecting some fluorescence when exposed to UV light. So this instrument works on the principle of emission of radiation. Here the UV light will be absorbed by our sample, and our sample biochemistry analyzer. This biochemistry analyzer is used for analyzing biochemicals, that is serum or plasma samples. We can do in vitro studies with the help of this instrument. We can bioanalytically measure how much the drug released into our serum or plasma matrix and all, and we can check the cholesterol level, creatinine level, and whatever the biomolecules we can analyze can be used, can be measured with the help of this instrument. Then the next instrument is distillation unit. This distillation unit is used to purify the sample. As the tap water is not suitable for analysis, we have to convert them into distilled water. We have to remove all the impurities as well as minerals. So that is why we have to use the distillation unit. Then the last one is, the next instrument is hot air oven. Hot air oven is used for drying our sample and as well as sterilization process. So with the help of this instrument we can dry our sample as well as stationary basis in case of TLC, and for these purposes and all we can use hot air oven. So there are many analytical instruments which have their specific purposes. We have seen only a few instruments, and we have